Good morning, everybody. Today we'll be talking about structure of cell membrane, intercellular junctions, and cell adhesion molecules. What is cell? As we have all learned in our lower classes, structural and functional unit of life is cell. A cell is the basic unit that is capable of performing life functions. So this is the structure of a cell. Have a quick glance, then we'll go in detail in our next coming slide. So this is a structure of a cell membrane. This is a lipid bilayer. In between we have proteins. We have different types of protein. The one which is spanning the entire thickness of the cell membrane, we call it as transmembrane proteins, and others are peripheral proteins which can face either the outer surface of the cell membrane or the inner surface of the cell membrane. We'll see it in detail in next few slides. Parts of the cell. So every cell in our body is covered by an outer layer, outer limiting membrane, which we call it as plasma membrane. Then we have a center nucleus, and surrounding the nucleus we have cytoplasm. Embedded in the cytoplasm we have different cell organelles. So this is a basic structure of a cell. The outer limiting membrane is the plasma membrane or the cell membrane. So this purple color structure is the nucleus. The yellow color one is the cytoplasm, and embedded inside the cytoplasm we have different types of cell organelles. Now, what is the structure of a cell membrane? We'll see it in a bit detail. Our cell membrane is a protective sheet of the cell. It helps in exchanging the material between the cytoplasm and the external environment. As I have explained you in the previous slide, the lipid layer of our cell membrane is arranged as a double layered, and the thickness is 7 to 10 nanometers. What is the constituent of our cell membrane? Our cell membrane is made up of 42% lipids, 55% are protein, and 3% is carbohydrate. So if I ask you which component is in highest demand, the answer will be proteins, which is 55%. So now this different scientists have proposed different cell membrane theories. So the latest and the not the latest, the most accepted one is Singer and Nicholson model, who proposed fluid mosaic model in the year 1972. Singer and Nicholson described the structure of cell membrane as a lipid bilayer with proteins embedded in it and which is free to move laterally within the membrane. So this is the structure of a cell membrane. Here we can see double layer of lipid. So this is yellow color ones. The round structure, the circles are the head, and tail is made up of two chains of fatty acid. So this is double layer of lipid. We can see head and a tail, and tail is made up of two chains of fatty acids. Okay, this is the more colorful picture which we have seen earlier also. This blue color one, the balls are the heads of the lipids, and this red color double chains are two chains of fatty acid, and the pink color ones are the proteins. Okay, embedded inside the cell membrane. As I said, either it can span the entire membrane of the cell, or either it can face the outer surface, or it can be embedded inside. So we'll see it. So this nature of this presentation, arrangement of the lipids in the cell membrane, we call it an uh, uh, our lipid has got two layers. It is arranged in double layer, head and tail. It has got head and a tail. This is one layer and this is two layer. That's why we call it as double layer. But what is the nature is heads are hydrophilic. Philic means loving, hydro means water. That means head are water loving, and tails are hydrophobic. Phobic means hating, hydro means water, so they are water hating. Tails are made up of two chains of fatty acid. They are water hating but lipid loving, and hydrophilic means they are water loving and lipid hating. So because they have dual nature, heads are hydrophilic and tails are hydrophobic, because it has got two natures, we call it as amphipathic nature. So our lipid, they are arranged in bilayer. This is one layer and this is the second layer, and head is hydrophilic and tails are hydrophobic, so we call this type of nature as amphipathic nature. So our lipid in the cell membrane is amphipathic in nature. Now, what is the meaning of fluid? What is the meaning of mosaic? Fluid means the presentation of phospholipids and the proteins, because they can move from side to side. Yellow color ones are the phospholipids and this gray color ones are the protein. because they can move from side to side. It's like a liquid. So the fluidity of the cell membrane is because of the presence of the phospholipid and protein. I can put it this way. Mosaic is because the proteins are arranged scattered. They are arranged in a scattered manner when we are viewing it from the above. So this presence of protein, arrangement of protein gives it a mosaic pattern. So we call this as fluid mosaic pattern. Fluid mosaic model, which was proposed in the year 1972 by Singer and Nicholson. So if I repeat, what is the fluidity of the cell membrane? It is contributed by the presence of phospholipid and protein which can move from side to side. Okay, the fluidity. And mosaic is because of the scattered proteins which are present, and when you're viewing it from the above, when you're viewing of a cell membrane from the above, it gives a mosaic pattern. So this is what I was saying. This flip-flop movement of the cell membrane. The cell can move laterally from one side to another side. The cell membranes are fluid structures. So movement of the cell membrane, as we are seeing in this picture, the blue color one, okay, one lipid you can see, it has come and tossed on the other side. That means this flip-flop movement of the lipids in the cell membrane. This we call it as flip-flop movement. So our lipids in the cell membrane could be of any type. It could be a phospholipid in nature, it could be a cholesterol in nature, or it could be a glycolipid. Okay. Glycolipid means lipid when it is bound to a carbohydrate, we call it as a glycolipid. Lipid when bound to the proteins, we call it as phospholipid, and this another one is cholesterol. So what are the special properties of this fluid mosaic model? First thing is that there are three important points to remember. First is the arrangement of phospholipid layer which is arranged as a double layer and amphipathic nature, and flexibility of the cell membrane. Our cell membrane is very flexible. Okay, and translational temperature, which is one of the important property which is exhibited by our cell membrane. What is the significance of our membrane fluidity? It is important for fusion of the membrane, that is fusion of the vesicle with cell organelles. It is important for diffusion of the new lipids and the new proteins laterally, so that these lipids and the new proteins, they are equally distributed at each corner of the cell membrane. Then another important function of fluidity of the cell membrane, or why the cell membrane should be fluid. It is because of diffusion of the proteins and other molecules laterally across the membrane in signaling reaction, and for proper separation of the membrane during the cell division. Now, factors which affect the fluidity of the cell membrane is length of the fatty acids, degree of saturation of the fatty acids, presence of cholesterol. This is an important MCQ. Cholesterol affects fluidity. What are the factors which are contributing to the fluidity of the cell membrane? It is the effect of temperature, and the second is lipid composition of the cell membrane. Now, what is the influence of temperature on the cell membrane? As we have seen, our lipid bilayer is amphipathic. So the hydrophobic chains of the fatty acid, they are highly aligned. So they give a very stiff structure. And whenever temperature is increased, our gel state, also called as crystalline state, it is getting converted to disordered liquid state. So the temperature at which our gel state is converted to liquid state or salt state, we call it as transitional temperature. And higher the transitional temperature, the fatty acid chain will be longer and saturated. So what determines the fluidity of the cell membrane? It is the degree of unsaturation which determines the fluidity of the cell membrane. Now, what is the lipid composition of cell membrane? So it is made up of phospholipid, at least one unsaturated fatty acid with cis bond. And as we have discussed, whenever there is increase in temperature, the unsaturated bond in the fatty acid, they lose their orderly arrangement. So cis configuration decreases and compactness is disordered. Now, what are the factors which are causing increase in the fluidity of the cell membrane? There are three important factors which causes increase in the fluidity of the cell membrane. The first is increase in degree of unsaturation. The second factor is increase in the length of the fatty acid. And the third is increase in the content of the phospholipid. These three factors, they increases the fluidity of the cell membrane. Now, what is the role of cholesterol? So cholesterol being a bulky molecule, and it is placed among the phospholipid. So whenever there is an increase in temperature, the phospholipids, they gain kinetic energy and the membrane start melting. So cholesterol, they pulls the fatty acids together, decreasing the fluidity. And whenever there is decrease in temperature, the fatty acid, they lose energy, they come very close, decreasing the intermolecular interaction, and cholesterol prevent this interaction, increasing the fluidity. Important point which I have highlighted in red: increase in temperature, cholesterol decreases fluidity, and decrease in temperature, cholesterol increases fluidity. So we can say that temperature and cholesterol, they have got inverse relationship. Now, cholesterol is a buffer and it tries to oppose the effect of fatty acid on fluidity to preserve membrane integrity within the ring. So cholesterol to phospholipid ratio is an important factor for membrane integrity. A lipid bilayer, it can be a phospholipid in nature, glycolipid or cholesterol. What is the function of a lipid bilayer? First is permeability barrier. So we have seen that because it has amphipathic nature, the lipid soluble substances can pass and water soluble substances cannot pass. Thereby lipid bilayer act as a permeability barrier. Next is fluid mosaic model. Lipids, it gives flexibility. It has got translational diffusion. There is a transition temperature at which gel state is converted to salt, and cholesterol to phospholipid ratio is important for maintaining the fluidity of the cell membrane. And we have said the lipids are amphipathic in nature. That is, head is polar, hydrophilic, and towards periphery, which is water soluble. And important function is it acts as a permeability barrier. Now we have different types of protein: integral protein and peripheral protein. Integral protein, also called as transmembrane protein, because they span the entire thickness of the cell membrane. Whereas peripheral protein, either they can be present inside or outside. If they are present inside, we call it as intrinsic peripheral protein. The example is enzyme. If they are present outside the cell membrane, we call it as extrinsic peripheral protein, and the example is cell adhesion molecule. So RBC membrane protein like spectrin and ankyrin are examples of peripheral protein. Integral protein, also called as transmembrane protein, they span the entire thickness of the cell membrane. They can function as ionic channels, carriers, ionic pumps, receptors, and they are antigenic in nature. Examples are RBC membrane proteins like glycophorin and band 3 protein. So, in few RBC membrane disorder like hereditary spherocytosis and eleptocytosis, the structure of the protein is disordered. So this is the arrangement of integral protein. The one which is I'm showing with the arrows, it is spanning the entire thickness of the cell membrane. Peripheral protein, if it is present inside, it is intrinsic peripheral protein. If it is present outside, okay, it is a extrinsic peripheral protein. So we are done with lipids, carbohydrate. We are done with lipids and proteins. Now we are into carbohydrate. The carbohydrate covering, which is covering the cell membrane, is a glycocalyx. It acts as a protective coat. It also helps in cell-to-cell adhesion, and it also have antenna, so it acts as a receptors. So what is the function of the cell membrane? Protection, anchoring one cell to another. It acts as a permeability barrier. It maintains intracellular environment. Okay, helps in maintaining cell volume and also potential difference between the nerve and the muscle. Now, cytoskeleton, there are three types of cytoskeleton: microfilament, microtubule, and intermediate filament. What is the function of cytoskeleton? They help in maintaining shape and structure of the cell membrane, the mobility of the cell, and mechanical support to the cell. It is provided by cytoskeleton. What are those? There are microfilament, microtubule, and intermediate filament. Microfilament is 8 nanometer in diameter. Microtubule is made up of two subunits. Okay, alpha tubulin, beta tubulin, it is also made up of gamma tubulin. Gamma tubulin is the one which helps in cell division. Okay, so these are the two protein subunits of microtubule which are they have highlighted in box. It is 25 nanometer. So intermediate filament which are third line, and intermediate filament is the one which is connecting nuclear membrane to the cell membrane. So now, difference between microfilament, intermediate, and microtubule. Microfilaments are made up of actin and myosin. Okay, and function is it is helping in contraction of the muscle, exocytosis, release reaction of platelets. Okay, they are present in microvilli, increasing the area of absorption, and they also act as focal adhesion complexes or molecular motors. Intermediate filaments they integrate organelles within the cytoplasm, resisting the rupture of the cell. So they act as a cell marker in few diseases. Microtubules, they are made up of tubulin. As I said, tubulin can be alpha, beta, or gamma. Gamma is the one which is important for cell division. Okay. Um, the assembly, there are arrangement and aggregation, disaggregation of microtubules is called as assembly and disassembly of microtubules. The function is they act as cytosolic guide rails. They help in cell division. They are also present at cilia and flagella. So this is the three important differences between microtubule, microfilament, and intermediate filament. So this is the structure of a microfilament. This is G-actin. This is tubulin. So the red one is alpha tubulin, and yellow one is beta tubulin. This is intermediate filament. Okay, which is important for integrating the nuclear components with the cell membrane.